Hello students, welcome back to Botany second year's class. So here we have started with the SAQs, and that also first chapter maximum SAQs we have finished, except this, that is the Munch mass flow hypothesis. This one only we need to finish it off, because this they may give or not in the supply exams. There is a doubt that they may give or not, but we have to go with this question also, so that all the questions from this chapter are going to be winded up, because from the first unit, that is the first to 6 chapters, that is transportation in plants and the mineral elements, that is the micronutrition and the enzymes, photosynthesis, respiration, and the growth hormones. Out of this, these are the 6 chapters that are there in the unit 1. So from this unit 1, we will get 4 SAQs. From which chapters they will take out these 4 SAQs, we cannot say. But maximum, how they will take out the 4 SAQs? First chapter, 1 SAQ. Second and third chapter, together we will get 1 SAQ. 4 and 5, together we'll get 1 SAQ. 6 alone, we will get 1 SAQ. Like that, 4 SAQs they will divide. So possibility is there for us to have the 1 SAQ from the first chapter. In the March, they have not done like this. But we cannot give that same assumption that this time also they will do the same thing. This time they may give the 1 SAQ from this chapter. That may be the somewhat easiest one. Okay, that may be the water potential answer. Otherwise, the plasmolysis. Otherwise, transportation is a necessary, it will explain. Otherwise, transpiration and photosynthesis a compromise. Explain. Both the answers, 3/4 answer is the same. So if you are preparing for one question, the another question, 3/4 answer, you will get it. Only photosynthesis you have to write down, and the last statement you have to change that in that answer. That much. So we can easily go with that, isn't it? And if we know the transpiration process, at least we can say the answer for this. That is essence of sap question also, because here cohesion, tension, adhesion, and the transpirational pull. So whatever we'll say in the transpiration, same thing we will say here also. So at least 2 marks, otherwise 1.5 mark you will get in this also, with the same concepts of what we learned in the transpiration is necessary, I will explain in that answer. So here we have to put that is these 3 questions are related with one topic in the chapter, that is the transpiration. If you know that concepts, you can easily give the answers for these questions. So you should be able to give that type of time and the brain maturity for yourself, that how to prepare for the IPE exam. And if we know the concepts, we can write the answers very easily. Now, the last question from this chapter, that is the explain the process of explain the pressure flow hypothesis of translocation of sugars in plants. This is the question, and the answer for this is the according to this, what we are saying, the pressure flow hypothesis of translocation of sugars. This is going to be done only by the phloem cells in the plant body. Phloem cells in the plant body, which transports the food material from the leaves to the other parts of the plant body. And this can be better explained with the Munch mass flow hypothesis. This was first given by Munch. The scientist name was Munch. Munch mass flow hypothesis. Munch mass flow hypothesis. So what he has given in this one, he has explained how the leaves are producing the food material in which form and how they are transporting from the leaves this prepared food material into the different parts of the plant body through the phloem in the upward and in the downward direction. That is both the directions. So always what we'll say, in the phloem, the molecules will move in the bidirectional, isn't it? Bidirectional means in both the ways, upward and in the downward direction. Whereas in the xylem, water moves only in the upward direction. From the roots to the upper parts of the plant body, not from the upper parts of the plant body to the roots. Never it comes in the negative direction, in the opposite direction. Only in the upward direction, water molecules will move through the xylem cells in the plant body. Okay, whereas in the phloem, how the molecules will move? Molecules will move in both the directions. That is in the upward direction as well as in the downward direction. That's the one here we have to say in the easiest manner how to write this answer. Okay, so here in which form the food is going to be prepared in the plant body, in the leaves by photosynthesis, in the form of glucose. And if you are going to say the food is prepared in the form of starch, that's wrong, because food stores in the form of starch, stored form. That means it is preparing in the form of glucose that can be stored for a long run. Then it should be in the form of starch. How we will store the things, isn't it? If we have to use now only, then we can keep the things in the normal container. If we have to store for the next month, otherwise the third month, for 3 months we need to store, means we will store the food, otherwise the whatever may be the stuff we have to store, that is the dry only, not the cooked ones. Okay, dry only. Then we will put in a airtight container, and each and every type, whenever we are using that, we won't use our hands because our hands may be wet, so the product may get spoiled. Otherwise, everyone knows about the lunch box, isn't it? No need to say about the things that are stored in the containers. If we know about the lunch box, whenever we are carrying the lunch box, we'll make the box to be dry. Why? If water is there, then the food is going to be spoiled within hours. By the lunch time itself in the summer, food will be spoiled if water is there in the container, in the lunch box itself, isn't it? If you are carrying the rice, otherwise the roti will become very soft. Okay, and we cannot able to cut it properly. It will be somewhat sticky because of the water that is present in the container, in the lunch box. The same thing here also for the plant. The plant also stores food in the form of starch for a long period of time. So the prepared food always in the form of glucose or sucrose. Okay, glucose or sucrose we can say, but stored food is always in the form of starch. Remember, but here we are not talking about the stored food material. We are talking only the freshly prepared food material in the chloroplast. that is in the leaf. Okay, so in that, the prepared food is in the form of the glucose, otherwise the sucrose form. So that only now we will see here. Okay, so through the phloem, the prepared food has to be transported in both the direction to the other parts of the plant body. When we are saying to the other parts, which part? Where food is needed for its development. Okay, and maximum amount of the food. That means maximum food is going to be shared, otherwise given to the root system, because roots cannot able to prepare their own food material. That much we know, because roots always present under the soil and brown in color, so they won't prepare the food. So food has to be given more in quantity to the roots always. There is no time limit for the food to be given to the, otherwise shared with the root system. Always it needs to be given to the roots, because without roots, plant cannot survive. If it says that I am only preparing food, why should I give to the roots? Then roots won't absorb water. There is no life for the plant body without roots. Without legs, can we stand? So to that type of people, if we are saying stand, that pinches us also after 5 minutes when we have seen them very clearly, because they cannot able to stand on their legs. They don't have the legs. Otherwise, those are the paralyzed, otherwise the polio affected. Then how can we say that stand on your legs? No, it's wrong. Na? Then we should apologize to them. Here also, the stem should not, the plant should not have the egoism that I am only preparing the food material in the leaves. So the leaves need to give the food always to the roots, that is always in the downward direction, the food transportation, and to the tip of the stem also, where the meristematic tissues are present, which has to divide and redivide and shows the growth in the plant body. So for that sake also, it needs maximum amount of the food, that is more number of the food particles are needed. So through the phloem, in both the directions, food need to be transported. And here we are saying leaves and the roots and the other parts of the plant body. Okay. So here, how this transportation, is this based on the concentration gradient? Obviously, because whenever the leaves are preparing the food material in the form of the glucose, that means glucose concentration is more, and in the roots, glucose concentration is less, which is going to be utilized continuously. Then from the high concentration to the low concentration, whenever molecules are moving, then which type of movement is that? High concentration of glucose to the lower concentration. So this is nothing but the osmosis, isn't it? From the high concentration to the low concentration through plasma membrane. Here, from the leaves to the roots. That means leaves are having the cell wall, plasma, epidermis, and the leaf cells, the cells we are talking. Okay, inside the leaves, it is having the mesophyll tissue. Mesophyll tissue means nothing but it is having its own cell wall, plasma membrane, cytoplasm, and the nucleus. So from this cell, the food needs to be moved to the other cell, that is in the root, till where it is also having the cell wall and the plasma membrane. So from this cell to this cell means through the plasma membrane. So that is nothing but with the osmosis. Okay, and here he has used Munch has used two different words instead of the leaves and the desired parts. Otherwise, roots are else another part of the plant body. Here he has done this experimentally. He has to show to the others. Okay, so he has used the words as the source and the sink. He has used the words as source and sink. What is meant by source? What is meant by sink? If you know one word meaning, another word meaning will come automatically. Source, where the food is going to be prepared. Where is food is going to be prepared? In the leaves. So here leaves are considered as the source, and the where the food is transported, that cell, that part is considered as the sink. That's it. Source and sink. If you know the basic concept, now you can write this answer with these two words. That's it. That's the answer what we are discussing here. See, okay, according to the pressure flow hypothesis, food is prepared in the food is prepared by the leaves in the form of glucose. What I said now, always food is prepared in the form of glucose. That's the one what they said. You can write in a simple words. I'll read this and again I'll say how to write in your own words. Okay, before moving into the phloem, the prepared food is converted into sucrose. Then only it can be easily transported from the one cell into the another cell through phloem. Okay. Then water moves from the xylem vessels into the adjacent phloem. Okay. Once food has to be transported, means food is always without water, isn't it? So with water only in the tube, everything can be transported. Otherwise, it can move, isn't it? So in a tube, if something blockages are there, solid particles, then how to clear off this? We will pump the water with a pressure over there so that the complete tube, the rust particles were cleared, isn't it? If you have seen your own buildings, the cleaning the water pipelines, then we can say. So just like a vacuum cleaner, with the vacuum also we can take out, with the water also we can pump. We can put the water in with a pressure, then the everything will be taken out into the adjacent phloem, thereby increasing the hydrostatic pressure in the phloem cells. Okay. Now the sucrose moves through the sieve cells of the phloem. These sucrose already present in the sink, that is in the so the sucrose is already present in the sink region, that is in the roots. Okay, is converted into starch, or it's a cellulosic form. Okay, somewhat complex form, thereby reducing the hydrostatic pressure in the sink cells, so that it won't give much more pressure in the sink cells itself. So those are becomes the empty spaces. Hence, the pressure difference created between the source and the sink, then it allows the sugars to be translocated from the source to the sink. This is the starch, otherwise cellulose is finally removed from the sink through the active transport. That's the one what we have to say. Okay, so till that we have to say this. So how to do this? How to write this answer? Now, start with the first point. Within 5 points, we can clear off this answer. How to write this answer? I'm making them to be there only. Okay. So explain the pressure flow hypothesis. That is of the translocation of sugars in the plants. This was given by Munch. The very first sentence, what we have to write? The pressure flow hypothesis was given by Munch to explain the movement of the prepared food material from the leaves to the other parts of the plant body. This is the first sentence, what we have to write over there. Next comes with so the prepared food will be always in the form of glucose. In the form of glucose in the leaves, which is going to be translocated through phloem through the phloem to the respective parts. Once it has to be transported through the phloem cells, immediately glucose is converted into sucrose form, through which it can be easily translocated through the phloem cells, phloem tissue. And once it is going to be translocated through the phloem, so which part of the phloem tissue, which part of the tissue, that is the sieve tubes. Through the sieve tubes, sucrose can be easily translocated. Once at that moment, when the sucrose enters into the sieve tubes, at that time, water is going to be transported from the xylem into the phloem cells, so that it makes the easy movement of the sucrose molecules through the sieve tubes to the other part of the plant body. Okay, now here this is food material is always present in the root cells. That is the root cells, or else in the brackets you can write it as sink. Before this also you can say, in this one we are saying, we are comparing the leaves as source and root as sink. So through the source, the sucrose and water molecules will move through the sieve tubes, and at that time already some amount of sucrose is present in the sink, which is going to be converted into the starch, otherwise its other form, that is the polysaccharides, okay, or else cellulose. It converts into the cellulose, so that it makes the easy transportation. That means the food can be transported from the high concentration to the low concentration. And with this, it is reducing the hydrostatic pressure in the sink cells. And like this, always the stored food, that is the excess amount of the food that is present in the sink cells, are going to be converted into the starch or cellulose, and finally removed further through the active transport. That's it. So in this, you have to draw the diagram which is having the source and the sink as the two containers. Okay, so the first one is the source dilute solution, and the second container, and these two are connected with the U-tube, not the YouTube channel. This is the U-shaped tube, okay, and having a disc-like structure at the bottom, and this is called as the A, and this is called as the B. Done. Now comes with the, and what is this? This is the A, container A. This is container B. So what it is having in the container A and the container B? So in the container A, it is having the dilute solution. Okay. And in the container B, it is having the concentrated solution. So no need for me to write the full form now. This is dilute. This is concentrated solution. Okay. Now, once we have seen the dilute and the concentrated solution, so from where to where the movement can be seen? From the concentrated to the dilute solution, the movement of the molecules can be seen now. So here from B to A, the movement of the molecules can be seen. Okay, once it is going to be moving from B to A, we have to show with one color. And now it is having another connection at the bottom. These two containers are having one more connection at the bottom. That is nothing but the so here through this one, the what again the movement of the molecules will be done from A container to B container. Then when this is just link that this is similar to the sieve tubes in the phloem. Okay, it is similar to the sieve tubes in the phloem cells. Okay, so now okay, similar to the sieve tubes in the phloem. Now what's happening when this is the A is considered as the sink and B is considered as the source. Source means leaves na. Here more concentration of the glucose which is converted into sucrose and traveling through the sieve tubes into the container A, where the dilute solution is present because of the already which was the food that is present in the sucrose, which is going to be converted into the starch, otherwise the cellulose in the sink. Okay, so there is an hydrostatic pressure, reducing the hydrostatic pressure, so which makes the movement of this glucose or sucrose from the B to the A, source to the sink. Okay, after this what happens, in the sink, once the concentration is more, then to reduce the one, it is again converted into the stored food is going to be transported through this into this as in the form of the starch, otherwise the cellulose particles, and otherwise it will be storing in itself only. So that is the sink, otherwise the storage tank, and the source is the leaves, otherwise the source. Okay, source is the leaf. Here also we can see leaf. Okay, so like this, and in this one we have to pour the water, pure water, so that water potential of the pure water is 0, which is very highest one. So once it is mixed up with the sucrose, then it will easily pass through the sieve tubes into the sink, where it is going to be collected. And once the collection will be done in a large quantity, then it will be converted into the starch, otherwise the cellulose form. Like that, always through to the source only, the transportation of the food particles can be seen. This much you have to write, and you have to stop this. Okay, so this is the Munch mass flow hypothesis, what we have to write. This one very rarely we will get in the supply exams. If you got also, if you write the answer like this, then it's the easiest one for you to score maximum marks isn't it? So now I'm not making you to lose a single half mark also in this paper. Now comes with the, now this was also, we have finished closing and opening of the stomatal apparatus and everything. Now we will start with the second chapter, that is micro elements. Okay, so in this, the very first question is the important one. Explain the steps involved in the formation of root nodules. Whenever we are saying this, formation of root nodules, we have to say this in the 5 steps. And if you draw the diagram, then done. 4 out of 4 only. So the root nodules are formed in the Fabaceae plants, Fabaceae family plants, okay, by the Rhizobium bacterium. For making the root nodules, it occurs in 5 steps. The first one, the Rhizobium bacterium, which is present freely in the soil molecules, were attracted by the sugars, amino acids, etc., that were released by the host roots. That's the very first point what we have to write over there. Okay, the freely living Rhizobium bacterium in the soil molecules were attracted towards the host roots by the release of sugars, amino acids, etc. Next one. Now, this Rhizobium bacterium will be accumulating. This is the root epidermis, and this is the root hair. And in this, the Rhizobium bacterium, the root hair is present in the soil molecules now. So the Rhizobium bacterium will be Rhizobium bacterium present freely in the soil molecules. This is the soil molecules. Okay, now what happens in the next step? Once the amino acids were released by the root hairs, what this Rhizobium bacterium will be attracted and starts accumulating at the tip of the root. And once they are accumulated at the tip, that is called as the colonize. Okay, that is called as colonize. They will be colonized at the root heads and multiply and attached to the epidermal cells of the root hairs. Now, these bacteria will be more at the root hairs. With this one, what happens to the root hairs? That's the third point what we have to say. Once they will be more at the root hairs, which makes the root hairs to curl. Till that root hair is straight now. Now the root hair shows a curl. Like this. This is by the accumulation of the Rhizobium bacterium near the tip of the root. Okay, once they are like this, what happens? Okay, now the bacteria starts entering into the root. Starts entering into the root by the disintegration of the cell wall of the root head. When they are entering into the root head and makes them and goes and settles in the cortex, so they will be looking like a string. that is called as the infection thread. Infection thread. Okay, so what happens once the bacteria enters into the root head by disintegrating the cell wall of the root head and makes its path till the cortex, then the way they are moving, that is called as the infection thread. Once the infection thread is produced by the plasma membrane of the root head, carrying the bacteria into the cortical cells of the host, which will stimulate the host cells to divide. So that's what I said in a easy language to you. Okay, then the host cells in the cortex cells, which makes them to divide and redivide by the food that is present in the cortex cells. Okay, so like that, many bacteria are present, and the other bacteria which cannot enter, they were present at the root hair only, which makes the nodule-like occurrence by the accumulation of the many number of bacteria, and that will be done by the formation of the ammonia. NH3, ammonia. Okay, so like that, the root nodules are going to be formed. This is the nodule. Okay, so in this, what happens, what they will do, this rhizobium bacterium convert the atmospheric nitrogen into ammonia, and this was given to the host nitrogen into ammonia, and this was given to the host cell. Okay, and instead of that, it is getting the shelter from the host cells. So this type of living, these microorganisms is called as the symbiotic association. Symbiotic association. Okay, so this is the one, and here one word was what I have written, that is the shepherd's crook. Once the root hair curls with the accumulation of the more number of bacteria, that is called as the shepherd's crook. Shepherd's crook. Okay, so whatever we have seen within the diagram, if you explain those points also, the answer will be coming to us, isn't it? That is also sufficient to get full score. That's what I'm saying. I'm not saying that don't read the matter. If you have not read the answer, otherwise if you cannot able to recollect it, then also it's easy for us to go and write this type of answer if you know the diagram and the labeling part. So in the root nodules, what we have to write first? The root nodules are formed only in the Fabaceae family in the plant kingdom. Which family? Fabaceae. Fabaceae family in the plant kingdom, where these root nodules were made by the rhizobium bacterium, which lives as a symbiotic, association with the host cells. Okay, this part. And now we have to say how the root nodules are formed. Root nodules are formed in different steps. This will be done in different steps as follows. The first one is the rhizobium the host cells. Okay, what we have to say, the root hairs of the host releases some sugars, amino acids, etc. into the soil molecules to attract the freely living rhizobium bacterium. Next one, next step. Second step, the rhizobium bacterium now accumulates at the tip of the root and starts releasing the enzyme to invade into the root hair. Invade means to go into the root hair. Okay, it digests the root hair cell wall and makes its easy entry, and some will be present at the tip of the root, which makes the colony of the bacteria. Okay, so once the bacteria enters into the root hair, it passes through the root hair and settles in the cortex zone of the root hair, that in the root, that is called as the infection thread. And once the accumulation of the bacteria, which makes the root hair to curl, that is called as the shepherd's crook, curling of the root hair with the rhizobium bacterium. Now, this rhizobium bacterium can absorb atmospheric nitrogen and converts it into ammonia. Okay, once this process was done, the ammonia was given to the host cells for its proper development. Instead of that, it is getting the shelter from the host cells. Okay, and now the bacteria will keep on multiplying in the cortex cells, and many number of bacteria were formed, which will make the ammonia formation in itself. So differentiation of the specialized nitrogen fixing cells occurs. The nodules are formed at the tip, and it establishes the direct vascular connection with the host for exchange of nutrients. So the other bacteria which are present outside, which will make the nodule-like occurrence at the tip of the root, that is called as the root nodule. Root nodule. That's it. This much we have to write over here. Next one is the next question, what we can expect in this chapter is the nitrogen cycle. Okay, so we will finish off with this question so that we can wind up 2 chapters. Okay, this you may get or may not get, but we have to learn. Only 2 important questions are there in this. So what is meant by nitrogen cycle? Give relevant examples. Nitrogen cycle means the cyclic movement of nitrogen from the atmosphere to the soil and from the soil into the atmosphere through plants, animals, or other microorganisms is termed as nitrogen cycle. Okay? Nitrogen cycle. Nitrogen cycle means the cyclic movement of nitrogen, atmospheric nitrogen, from the atmosphere into the soil. Okay, from the soil into the plants, animals, or microorganisms, and that is called as the nitrogen cycle. Okay, So once this nitrogen cycle, it is having the nitrogen cycle is having 5 different steps in it. That is the first one is the nitrogen fixation, nitrogen assimilation, ammonification, nitrification, and denitrification. Now comes the first one, that is the nitrogen fixation. Okay, in the nitrogen fixation, what we have to say, nitrogen, atmospheric nitrogen is going to be fixed in the different organisms, that is in the plants, otherwise directly in the plants, otherwise in the soil molecules in the form of ammonia by 2 different methods. Okay, nitrogen can be fixed into ammonia in plants or in soil molecules. So this is called as the nitrogen fixation. So this nitrogen fixation can be done by a biological method and the biological method. A biological method, that means naturally. Naturally, from the outside atmosphere, nitrogen is converted through the lightning energy, otherwise by the combination of the different nitrogen into ions in with the excess amount of the water molecules, it is converted into the NO, NO2 and NO3 forms. Okay, NO3 forms. This is the nitrogen oxides. Nitrogen is converted into nitrogen oxides. Nitrogen monoxide, nitrogen dioxide, otherwise nitrogen oxide, NO3. So like this, it can be converted by the lightning energy that is present in the outside atmosphere during the rainy season, otherwise by the availability of more number of the water molecules with the nitrogen gas. N2 plus H2O gives NO. Okay, that is 2 molecules of NO. That was over. Now here 2 molecules. Then 2H2 is going to be released. Like this. Like this, the conversion. Now this 2NO is again combined with the more amount of the water molecules and forms NO2 form or NO3 form. Anything. Like that, the nitrogen is converted into nitrogen oxides. Otherwise, when through the industries, that means from the industries, whenever the waste materials are going to be released into the outside atmosphere by the combustion, otherwise the forest fires and any other way, that is the power generating stations, in the atmosphere, nitrogen oxides are going to be released. Those are also converted into this form, and that goes directly with the rainwater into the soil molecules. Okay, that is going to be fixed in the soil. That is the a biological method. That means where no other living organisms are making the help in converting the atmospheric nitrogen into ammonia, otherwise the nitrogen oxides. Once it comes with the biological method, that is the living organisms. With the help of the living organism, that is especially the prokaryotic organisms, that is the microorganisms, which will make the conversion of the nitrogen, atmospheric nitrogen into ammonia. This occurs with the rhizobium bacterium, nitrifying bacteria, otherwise the rhizobium bacterium, and the cyanobacterial members, which will convert the atmospheric nitrogen into ammonia by absorbing them and converting it into in the body with the help of the enzyme nitrogenase, which is capable of the reduction of the nitrogen. Okay, so that is present in the prokaryotic organisms. That is the most important one. And in this, the free living nitrogen fixing aerobic microbes are the Azotobacter and the Beijeringia, and the Rhodospirillum is an anaerobic bacteria. Okay. Now, like that, if you remember this, you can write, otherwise no need. And the cyanobacterial members, what will say? In that nostoc, you can say, example, nostoc as the cyanobacterial members and the rhizobium. You can wind up. So that is coming into the directly into the soil. That is a fixation. Next comes the assimilation. Assimilation means that is going into the plant body and it is converted into the different forms in the and stored in different parts of the plant body. That is called as the assimilation. So the nitrites and the ammonia, it can be in the soil molecules. The nitrogen now is present in the form of the nitrates or the ammonia, which is absorbed through the soil water by these plants and can be converted and the stored in different parts of the plant body in the form of the enzymes, otherwise the pigments, hormones, nucleic acids, proteins, amino acids. In different forms, this nitrogen is going to be fixed in the plant body. And now this it is in the form of the organic form of the nitrogen. When the plants are eaten up by the grazing animals, and these grazing animals, in this one, this nitrogen enters. And once the carnivorous animals will take out these grazing animals, and in their body, this nitrogen will go. Like that, from the herbivores, the fixed nitrogen in the different forms is going to be exchanged from this organism into the primary, that is in the carnivores, otherwise in the human beings also. Like that, it is going to be in the human beings. It can be coming into through the plant part, that is through the leaves, fruits, vegetables, otherwise through the animals, herbivorous animals, that is a goat. Okay, like that. Through them, we will be having, that is the nitrogen assimilation. So now this is completely converted into the organic form, and it is the absorbing nitrates, ammonia, and chemically bonding with the nitrogen in the different organic materials. That is the one what we can say in the nitrogen assimilation. Now comes the ammonification. In the ammonification, what we have to say, again, it needs to be converted into needs to go back into the outside atmosphere. Now, how it will go? It has to be decomposed by the decomposition of the organic nitrogen by the dead organisms into ammonia. Now, the organic nitrogen is going to be converted into ammonia. Okay, by the specially made the microorganisms. That is, by the death of these organisms only, this can be done. That is the ammonification. Ammonification means decomposition of the, otherwise conversion of the organic nitrogen into ammonia through the dead parts of the plants, animals, or human beings by the microorganisms is called as the ammonification. Okay, so here the bacterial organisms are the most important ones. Those are called as the bacillus type of the microorganisms. And the fourth step is the nitrification. In the nitrification, what it happens, the ammonia is going to be converted into the nitrates by the nitrosomonas and the nitrosococcus bacteria. And the nitrites are again oxidized into the nitrates. Okay, that means ammonia is oxidized into the nitrite and the nitrate by the nitrosomonas and the nitrobacter respectively, so that it's going into the outside atmosphere, and by the denitrification, it is again converted into the atmospheric nitrogen through the Pseudomonas, otherwise the Thiobacillus bacteria. Like that, completion of the nitrogen cycle occurs, and you have to draw this flow chart. That is the nitrogen cycle. With this, we have completed the 2 chapters SAQs, and we will see the 3rd chapter SAQs, which are most important. And stay tuned to the same channel, same time for the next chapter SAQs and the VSAQs. Right? Thank you.